I would like to start introducing what my lab has been doing or has been up to in the last year and what are we working on. So this title is a little bit out of context, but I will talk to you a little bit about the crux that we are facing and how we are planning to solve it. And I would like to start with that our lab historically has been interested in hair cells. I came from a hair cell lab as a postdoc, and I'm fascinated by the cell. And then particularly fascinating is that we know so little about the cell. If you compare hair cells with any other cell in the body or any other sensory cell, it's probably one of the cell types we know the fewest about them, particularly how they convert mechanical stimulation into electrical signals. And I'm glad that actually one of the postdocs in our lab holds up this, this classic or historic interest in hair cells, and Mica Herget is actually pursuing a project that started a while ago with looking at a hair cell specific protein that lots of people in the developmental biology field have been using as a marker for chicken hair cells. And Mica has used antibodies against this protein, which turned out to be otoferlin. And she has identified quite a number of interaction partners to this protein, and she's currently interested in CAM kinase 2, which could play a role in regulating the molecular function of otoferlin. So her general interests are the molecular mechanism acting on otoferlin and the molecular function of otoferlin. And this is a collaboration with more and more with Tony, because this work goes more and more into very likely physiology at some point. And Mike is collaborating with Felipe and with people from the Garner lab on this study. Mica has been actually very successfully obtained funding for her research. She's independently funded with a 3-year fellowship from the German Science Foundation that helps her enormously to be independent for doing this kind of research. Lars has been in our lab for a while, and Lars is interested in a couple of things. And one of his interests is actually a hair bundle protein. So we go from a bottom to top to the hair cell, and we do it very quickly because we don't have a lot of projects working on molecular mechanisms of hair cell function. On the other hand, we have an R1 grant that actually funds this research, and then I'm now trying to figure out how to pursue this research with what we're having. But I think what Mica and Lars are doing is actually quite promising, and I think we might be able to sustain this line of funding. Lars lately has become a little more interested in mice and actually in doing quite a lot of hearing tests. And with the help of our core facility, Jerry Popelka and also John Ogalai and people involved in that, Lars has been pursuing a number of projects checking hearing in mice. And in a collaboration with Ricardo Dolmetsch's lab, we have Lars has been looking at a mouse model of a human autism disorder. And in this mouse model, surprisingly, because the humans that have this microdeletion in their genome, the humans don't have any hearing disorder, but the mice are profoundly deaf, and they also have balancing defects, which points perhaps to a gene that was hit by accident in this kind of study. And then we have some very vague idea how to pursue this gene, and Megan Ely, a postdoc in the lab, has become interested in this recently as well. So this poster, if you're interested, is up in the hallway. One thing I'd like to mention is all the posters that we are presenting is unpublished work. We have been sort of in a mode of getting new people into the lab and also trying to finish out other projects that we are working on, which is a quite interesting situation. We never had that situation where all the posters are unpublished, which I think is probably a lot of work in the future to write up all these papers. What is interesting also is that if you're interested in regeneration of hair cells are not the main focus. The hair cells actually die. And then what is more interesting is supporting cells. And Supporting cells are very interesting cells. They haven't been investigated a lot. People haven't paid a lot of attention to supporting cells classically, but I think if you're interested in regeneration, these are the cells that are left over. These are the cells you have to work with to regenerate hair cells. And one organism or one group of animals that readily regenerates hair cells are chickens or birds. Birds and fish regenerate hair cells. And Mirko is interested in the mechanisms of avian hair cell regeneration and also in this mechanism in chimeric chick and mouse ears. And he's currently working on creating a tool, a time-lapse imaging tool, where we can put chicken utricle in culture and observe them over time. And because we cannot simply observe cells with a microscope and see where, for example, the nuclei of individual cells go and what the fate of these cells is, he's actually developing a couple of viral vectors to label supporting cells and also to label the process of differentiation, namely when these supporting cells decide to differentiate into a hair cell. And then his early efforts in this regard are presented in his poster. And he's also working together with a high school student on a novel microfluidics-based chamber that we can use to study tissues for an advanced amount of time, hopefully up to 2 weeks in a microscope. And this work that I'm talking about now is funded by a second NIH grant that focuses on mouse inner ear progenitor cells. And Robert is working on one of the cruxes that I mentioned earlier. And this is, the question that he's asking is, what are the transcriptional profiles of different otic progenitor cells? And then one thing we figured out quite recently is that what we thought is a cell type, an otic progenitor cell, that is, for example, localized in the mouse otocyst, which is the structure that later on gives rise to the inner ear, that this otocyst doesn't contain a very specific otic progenitor cell, but it contains populations of different cell types and different progenitor cells that can give rise to all the auditory and vestibular neurons, for example, or they give rise to non-neural structures, or they give rise to perhaps the sensory epithelia. And the prosensory epithelia progenitors are the cells that we are highly interested in. So Robert uses a technology that was developed in this building in Stephen Quake's lab, which is a fluidigm-based single-cell analysis system. And then some of you have seen this new apparatus that was delivered into the imaging core recently. This is the machine where we can do this kind of research. This is a profile of otocyst-derived PAX2 positive cells. All of these cells express PAX2. They have been sorted with a cell sorter, and this is the PAX2 expression in these cells. These are embryonic stem cell generated cells that express PAX2, here shown by a GFP reporter, and here shown, these are the negative cells that we also sort. So we got the positive cells and the negative cells that have been guided in vitro to become inner ear cell types. And these are the cells we have used in a recent paper about 2 years ago, Kazu Oshima's work, to actually generate inner ear hair cell-like cells. And what we see in these populations is that another ear marker, PAX8, is actually nicely co-expressed with PAX2. So these individual bars represent individual cells. So this cell here expresses PAX8 and PAX2. And what you can see is in our in vitro generated cells, we have also a few cells that express these two markers, but not all of them. This explains perhaps one of the issues we have been dealing with which is efficiency. The efficient generation of inner ear cell types from embryonic stem cells is one of the problems that we are having right from the onset when we use this protocol. Here's another marker. FBX2 is a marker that was found in a microarray study by Biron Hartmann in the lab, and I will show you a little bit more about this in a second. And FBX2 is nicely co-expressed with PAX2 positive cells, not with all of them, because it turns out FBX2 in the otocyst is only expressed in the very ventral region of the otocyst, at least on the transcriptional level. That's what we think. And again, only a few cells co-express these ear markers. So the crux that we are dealing with is that cells that we generate from embryonic stem cells are representing inner ear cells, but the cells that are truly representing the cells that later on become prosensory cells is really a small proportion of the cells that we are dealing with. And then we need to characterize these cells in much more detail. There's always a good and a bad. The bad I told you about. The good side, though, is that other markers that label other cell types, for example, PAX6, which is expressed in neural progenitors and also is a retinal progenitor marker. This marker is not expressed in the otic vesicle side, one outlier here, and also in the cells that we think are going toward an otic lineage. This marker is absent, whereas it's present in the surrounding cells. So we have been on the right track, but we just have to increase efficiency. And knowing what the efficiency is actually one of the big, for us it's a breakthrough, knowing what we are dealing with, because now we can solve it. Biron is also involved in this work. Biron is interested how otic-specific gene regulation is achieved at the transcriptional control level. And he has been putting one and one together in the last year, looking at microarray data previously generated in the lab and also generated by him and Roman Lasky, an MD scholar, a visitor from Switzerland. And he has found a number of genes that are truly otic markers. And then he has, on his poster, he has actually identified the crux again. So embryonic stem cells can be used for quite a lot of good things. But the crux is that there are two critical barriers. One is that we do not have a lot of markers that are truly specific for the ear. And the other one is that we don't understand what the genetic regulation of these markers is. What really defines, and that goes also into Robert's work, what defines an otic progenitor cell? What regulates the genes that make this progenitor cell? And so Biron has been looking at some of these markers, and with the help of Mirko Scheibinger and Robert Dorothy as well, they are pursuing quite a number of genes that are truly specific otic markers. This is FBX2 again, and this is just a whole mount immunostaining done by Biron that highlights that this protein and this gene as well is highly specifically expressed in the developing inner ear. And we haven't had these markers before. So they are of great use for the future, and this is what we are currently working on. Kazu Oshima has been the cleaning lady of the lab. He's pursuing all the unfinished projects. And then one unfinished project that he's working on is actually quite promising. And it might be something for building a career. So Kazu is currently thinking about making some of this part of what he's doing in the future. And what he's interested in is what controls postnatal neurogenesis in the cochlear nucleus. This goes back to some studies done by Sam Most in his research time, where he looked at survival in the cochlear nucleus of neuron. We are looking at neurogenesis, and Kazu found also, together with Sam and in a collaboration with Alan, that there's an input-dependent survival of these progenitor cells that give rise to new neurons in the cochlear nucleus. And when you take away one cochlea, so you remove cochlea with surgery, you can actually see that the number of these cells, which we call progenitor cells or sphere-forming cells, it decreases, and you can see more of this on his poster. Milica is working using the in vitro guidance system to actually recapitulate mouse hair cell phenotypes in IPS-derived hair cell-like cells. IPS cells are cell types that can be generated from any cell in the body, and Yamanaka just got a Nobel Prize for this technology, so you probably all heard about this technology. What we principally do, we take a skin cell or an embryonic fibroblast from a mouse, let's say a Shaker 2 mouse, which has short hair bundles. And this is a heterozygote or a wild-type comparison. And Milica is looking actually at the hair bundles of hair cell-like cells generated from these mice that have this very specific phenotype and compares it with heterozygotes. Megan, Jörg, and Mirko are working together on actually generating induced pluripotent stem cells from patients with mutations in specific genes, or also from patients who have susceptibilities. And this is the process that they're using. This is the Yamanaka invented process to actually generate stem cells. And Megan's own project is actually asking the question whether susceptibility to ototoxic insults can be observed in inner ear cells generated from IPS cells. And she's pursuing this initially on the mouse side, but also then later on patient-derived cells. How can we do this in patient-derived cells? There's one fundamental project that is where everybody is circulating around. It's run by Mohammad Ronaghi and his wife, Marjan. And they have actually developed a protocol to guide human embryonic stem cells to differentiate into inner ear cell types, including cell types that look pretty much like hair cells, although we haven't done any functional analyses or functional studies on these cells. There are a couple of roadblocks that we had to overcome, and then I encourage you to go to this poster because they have really generated human embryonic stem cells that are transgenically marked and that have reporters, so we can actually see whether hair cells develop in a culture system. This technology, as I mentioned, is being used by the group around Megan, Mirko, and Jörg. And Megan and Mirko are interested mainly in, for example, the hair cells, the hair cell-like cells that this study can hopefully produce. And they look into susceptibilities, and Mirko looks actually into the same mutation that Milica investigates in the mouse, but he is focusing on human patients that carry this mutation. Jörg is interested in the supporting cells, and he's interested in connexin 26. And as you have heard already earlier, connexin 26 is the main gene mutated in a lot of hereditary hearing losses, and we are interested in studying this in patient-derived cells. Oops, that's my own timer. Okay, I will hurry up. Jörg also has a project that deals with direct conversion of mouse fibroblasts into otic cells, and he does this by applying a battery of transcription factors. I think he's up to 30, and he looks whether it's possible to convert a skin cell directly into an inner ear cell, or a skin cell directly into a hair cell. Although we have to figure out which is the better process. And I'm more in favor right now to converting a skin cell into a progenitor cell, which very likely then leads to repair. Let's imagine you can do this in a damaged organ of Cordy. So this is the whole group. These are the 3 NIH grants by which this is funded. Several of these guys are funded through scholarships. Biron just got a very good score on his F32, and he will probably know on Monday whether he gets money. Mirko is funded by a 3-year grant from the German Science Foundation. Mica is funded by them as well. And Jörg is currently having a Dean's Fellowship. And we have a number of collaborators. I have to thank Tony, who has been taking care of a project that we just submitted with a former postdoc, Jessie, who is in St. Louis and doing well. She has not been converted to a Bifang-like thing. Tony works with Mica, we work with Felipe, we work with Jerry. We have a lot of projects and a lot of ideas shared and going on with Ellen. With Sam, we have a shared R1 with Richard Smith in Iowa, which was the product of his visit 2 years ago to Research Day, working with Tom Friedman at NICD, and Sheikh Riadusin in Lahore in Pakistan to actually get our hands on patients that have this specific mutation. Thank you.